Pleasant day, everyone. I am Jeffany Dimutya, and since we already tackled the previous parts of the our topic, which is the biotechnology, let us now move on to the last and third part of our topic, which is the cloning. Is cloning? Cloning is the process of creating a genetically identical copy of an organism, cell, or DNA sequence. This can be done through various methods, and the result is an entity with the same genetic makeup as the original. So, the most popular form of cloning is Dolly the sheep. So, we will tackle that as we dig deep in our topic. So the idea of cloning animals was first suggested in 1938 by German embryologist Hans Spemann, or also called the father of modern embryology, who proposed what he called a fantastical experiment. It is, as what you can see in the slide, it is in simple terms, removing the nucleus from an egg cell and put in its place a nucleus from another cell. Experiments show that cells have the ability to develop into a whole organism, which was a huge deal in understanding how life works. It's like finding out a tiny seed has the potential to grow into a whole tree. It was 14 years before technology advanced far enough for anyone to take up Spearman's challenge. But in 1952, there were two American scientists that take up Spearman's challenge, which were Robert Brace and T.J. King. So their experiments involve the sucking of the nucleus from a frog. And this experiment did not work when done this way, but partial success was achieved 18 years later by the British developmental biologist John Gordon, who in 1970 inserted nuclei from advanced frog embryos rather than adult tissue. The frog eggs developed into redpolls but died before becoming adults. For 14 years, nuclear transplant experiments were attempted without success. Technology continued to advance, however, until finally in 1984, Sten Weladsen, a Danish embryologist working in Texas, succeeded in cloning a sheep using a nucleus from a cell of an early embryo. This exciting result was soon replicated by others in a host of other organisms, including cattle, pigs, and monkeys. Researchers thought that once embryo cells starts differentiating, like becoming specific types of cells, you can't use the nucleate to clone whole organism. But Kate Campbell had an idea. What if the egg cell and the donor nucleus need to be at the same stage in the cell cycle? Like egg needs to be ready for the nucleus to work properly. His understanding that cells don't divide until conditions are appropriate, just like a washing machine checks that the water has completely emptied before initiating the spin cycle. So the cell checks that everything needed is on hand before initiating cell division. So Campbell reasoned maybe the egg and the donated nucleus need to be at the same stage or need to be ready in order to function properly in the cell cycle. Because in previous experiments it was not successful. So, in Campbell's perspective, in order to have a successful experiments of cloning, is the cell must be or they must be in the same stage, which is the ready stage in order to function properly in the cell division or in any steps that helps in the progress of the cloning or the results of cloning. This proved to be a key insight. In 1994, researcher Neil First and in 1995, Campbell himself, working with reproductive biologist Ian Wilmot, succeeded in cloning farm animals from advanced embryos by first starving the cells so that they paused at the beginning of the cell cycle at the G1 checkpoint. Two starved cells are thus synchronized at the same point in the cell cycle. The Wilmot slump, it is the transferring of the nucleus from an adult differentiated cell into an unclated egg and allow the resulting embryo to grow and develop in a surrogate mother, hopefully producing a healthy animal. So Wilmot removed mammary cells from the udder of a 6-year-old sheep. The origin of these cells gave the clone its name Dolly, after the country singer Dolly Parton. The cells were grown in tissue culture and some frozen so that in the future it would be possible with genetic fingerprinting to prove that a clone was indeed genetically identical with the 6-year-old sheep. So let's take a look at this picture. So there is this extraction of mammary cell and egg cell from different sheeps. A mammary cell, a cell from the mammary gland or breast tissues of a female mammal. In Dolly's case, it was cell taken from the sheep's udder. Egg cell is a female reproductive cell like an ovum. In cloning, the nucleus is removed from this egg cell and replaced with a nucleus from another cell, with or a mammary cell. So as what I had said earlier, there is this extraction of mammary cells and egg cells, were then surgically combined and then the mammary cells inserted inside the covering around the egg cell. Wilmot then applied a brief electrical shock. A neat trick, this causes the plasma membrane surrounding the two cells to become leaky, so that the contents of the mammary cell passes into the egg cell. The shock also kickstarts the cell cycle, causing the cell to begin to divide. As what you can see in the picture, so there is this combination of mammary cells and egg cells, and then there is this electric shock that causes the cell to begin in division, cell division. So that's how the Wilmot's lab is done. So Wilmot's successful cloning of fully differentiated sheep cells in a milestone event in gene technology. Even though this procedure proved inefficient, only one of 277 trials succeeded. It established the point beyond all doubt that cloning of adult animal cells can be done. In the following 4 years, researchers succeeded in greatly improving the efficiency of cloning. Seizing upon the key idea in Wilmot's experiment to clone a resting stage cell, they have returned to the nuclear transplant procedure pioneered by Briggs and King. It works well. Many different mammals have been successfully cloned, including mice, pigs, and cattle. Also, transgenic cloning can be expected to have a major impact on medicine as well as agriculture. So what is transgenic cloning? So transgenic cloning is when you take a gene from one species like humans and put it in another species like, for example, sheep, and then you clone the animal so it has a new gene in every cell. Let's take for example a sheep that have been recently been genetically engineered to secrete a protein called alpha-1 antitrypsin, helpful in relieving the symptoms of cystic fibrosis. Into their milk may be cloned, greatly cheapening the production of this expensive drug. So in simple terms, it's to take a gene from one creature like human, put it in another creature like sheep, and then clone the creature so that it has the new gene in every part of its body, and now the sheep makes something like a protein that humans need for medicine. So in that case, we can't really deny the fact that cloning has a big impact in our life, our daily life, as well, especially in our medicine and agriculture. So now let us move on to what is a stem cell. At the dawn of a human life, a sperm fertilizes an egg to create a single cell destined to become a child. As development commences, that cell begins to divide, producing small whole of a few dozen cells. At this very Carly point, each of these cells is identical. We call these cells embryonic stem cells. Each one of them is capable by itself of developing into a healthy individual. In cattle breeding, for example, these cells are frequently separated by the breeder and used to produce multiple clones of valuable offspring. By this simple definition, we can now know what really is stem cell. So there is this fertilization of the egg cell and the sperm cell, and also the division that